I'm Dr. Reena. I'm working as a lecturer in Government College for Women, Sector 14, Panchkula. Today, I'm going to discuss about penicillium. Its common name is green and blue mold. First of all, we'll discuss about the systematic position of penicillium. It belongs to division Eumycota, class Plectomycetes, order Aspergillales, family Aspergillaceae, and the genus is Penicillium. So in this, we'll discuss about its occurrence and habit, and then vegetative structure, and then after that, I'll explain you about the reproduction, asexual and sexual reproduction. So first of all, we'll discuss about its occurrence and habit. So Penicillium includes 136 species and it has saprophytic mode of nutrition. It grows on decaying vegetables, fruits, meat, rotting plants and animals. As it takes food from the dead organic matter, it releases enzymes which convert the complex organic molecules into simple. In this way, the digestion takes place outside its body, and a few species are parasitic also and they cause diseases. Some of the common species are Penicillium italicum, Penicillium digitatum, Penicillium notatum, Penicillium chrysogenum. Now this Penicillium italicum causes blue mold of citrus fruits in storage. Penicillium digitatum causes green mold of citrus fruits in storage. Penicillium notatum and Penicillium chrysogenum, they have antibiotic properties, so they are used to produce penicillin. There are some other species also like Penicillium expansum. It causes rotting of apple, pear, grape fruits in storage. And Penicillium purpurogenum cause spots on the printing papers and the books. Now we'll discuss about the vegetative structure of penicillium. You can see here in the diagram that penicillium, its plant body is known as mycelium. It grows superficially or penetrate deep into the substratum, and sometimes it forms a compact mass called sclerotium. Now this mycelium consists of hyaline, pale or brightly colored, tubular, branched, septate hyphae, and these septa are perforated, or you can say there's a pore in the septum. Because of that, the cytoplasm of these cells is in continuation with each other, and the cells are thin walled. They are uni, bi or multinucleated. For example, Penicillium vermiculatum, the cells are uninucleate. They have granular cytoplasm. Organelles are there and the oil globules are also present. Now we'll discuss about the reproduction. In Penicillium, the reproduction occurs by both the methods, asexual and sexual method. First of all, we'll discuss about the asexual reproduction, and this takes place by the formation of conidia on the conidiophores. Now we'll discuss about the formation of conidiophores and the conidia. You can see here in the diagram that conidiophores are developed from the vegetative cells only, the vegetative cells give rise to septate hyphal branches, and these branches grow vertically and called conidiophores. Now, these conidiophores may be monoverticillate or biverticillate. So you can see here the first type is monoverticillate, means the conidiophores are unbranched. And on the tip of conidiophore, which is unbranched, a cluster of sterigmata are present. These are bottle-shaped structures. And second, you can see here biverticillate type conidiophores, which are branched. So branching may be twice or thrice or more. So these branched conidiophores, their ultimate branches are called medullae, and each medulla bear number of bottle-shaped sterigmata. So you can see here that each branch ends with the medulla, and each medulla bear cluster of sterigmata, and each sterigma is long bottle or flask-shaped structure which bear the conidia in chains. And in this way, you see here that the conidia are present in chains. Now I'll explain you that how conidium is formed. So you can see here the conidiophore is a vertical branch which is rising from the vegetative cells, and at the tip of this conidiophore there is present a cluster of sterigmata. Now we'll consider that how the conidium is formed from this sterigma. Now you see here that a swelling is formed at the tip of sterigma, and this swelling increase in size. Later on, the nucleus of the sterigma divide into two, and one of the nucleus passes into the swelling, and this swelling becomes separate from the rest of the sterigma by septum formation, and later on it develops a wall around it and conidia formation take place. Now this conidium is pushed forward and the next swelling is developed and leads to formation of next conidium. Now these conidia are formed in chains in basipetal manner, that is the older towards the apex and younger towards the base. And if we see the structure of conidium, conidia are generally blue-green or pale in color. They are globose or ovoid in shape, and conidium is non-motile, uninucleate, and has two wall layers. The outer wall is thick, it can be smooth or sculptured, and inner wall is thin. These conidia is taken by wind, and when they fall on the suitable substratum with suitable conditions of temperature and moisture, then they germinate. And each conidium germinate by giving out one or more germ tubes which grow and form mycelium. If the conidia are placed in the saccharine solution under certain conditions, then they form buds like yeast. This is called tortula condition. And like yeast also, it induces alcoholic fermentation. So this is about the asexual reproduction, how conidia are formed on the conidiophores, and how they germinate to produce a new mycelium. Now we'll discuss about the sexual reproduction. Sexual reproduction and the production of fruiting body, that is Cleistothecium or ascocarp, has not been reported in most of the species of Penicillium. But it has been found in some of the species of Penicillium. Most of the species of Penicillium are homothallic. For example, Penicillium vermiculatum. And mode of reproduction may be isogamous, oogamous, or gametangial contact type. So isogamous type of reproduction was found in Penicillium bacillosporum, and oogamous type is found in Penicillium vermiculatum. Now we'll discuss about the development of sex organs. The female sex organs are escogonium, and the male sex organ is antheridium. Now you can see in this diagram that how escogonium and antheridium are developed. So you can see here, escogonium arises as a lateral outgrowth from any vegetative cell. In the beginning, it is uninucleate, but later on, it grows and elongates. Nucleus also divides repeatedly and forms 32 to 64 nuclei. So it becomes a long, tubular, multinucleate, non-septate structure, which is slightly curved at the apical end. So you can see here in the diagram that this elongated female branch, or you can say this forms a young escogonium. And now the antheridium also developed as an antheridial branch, which is close to the escogonium. So either the antheridial branch arises from the same mycelium or the other mycelium. This branch, or you can say the antheridial branch, is uninucleate. It elongates and coils around the escogonium. You can see here in the diagram that how it is coiled around the escogonium. And finally, it cuts off as a uninucleate antheridium by the formation of septum. Now, you see here that it is uninucleate antheridium. Now, the next step is plasmogamy. The two sex organs, escogonium and antheridium, are in close contact with each other. Now, during the sexual fusion, the common wall between the two, that is escogonium and antheridium, dissolves, and the protoplast, except the male nucleus, migrate into the escogonium. So it is very interesting to see here that nucleus does not migrate into the escogonium, only the protoplasm migrate into the escogonium. So this fusion of protoplasm of the escogonium and antheridium is called plasmogamy. So you see here in Penicillium, the male nucleus remain in antheridium, and later on it might degenerate, but in this case the antheridial protoplasm stimulate the escogonium to develop further. Now, what happens after plasmogamy? That is a fusion of protoplasm of the escogonium and antheridium. Now, after the plasmogamy, the male nucleus may degenerate in the antheridium. So, as I told you, that antheridial protoplasm stimulate the escogonium to develop further. Now you can see here, after the plasmogamy, the escogonium divides into large number of cells arranged in a row. So you can see here in each cell there is a pair of nuclei present, and you can see here that some of the cells in the middle of escogonium develop one or more lateral escogonial initials, which form branched escogenous hyphae, and it is binucleate. The terminal cell of the escogenous hyphae, or I can say this branch, function as Escher's mother cell. You can see here in the diagram that Escher's mother cell increase in size, that is the terminal cell of the escogenous hyphae. Now what happens that the two nuclei in these Escher's mother cell fuse, and this fusion of the two nuclei in the Escher's mother cell is known as karyogamy, and this leads to formation of diploid nucleus. Now this diploid nucleus undergo further divisions. First, meiosis takes place, because this diploid nucleus, when it undergoes meiosis, it produces haploid nuclei, and later on mitosis takes place, which leads to formation of 8 haploid daughter nuclei. Now what happens that the cytoplasm is collected around each nucleus, and in this way 8 ascospores are produced, and in this way you can see here the asci are spherical or pear-shaped, which bear or which contain 8 ascospores which are spherical in shape. Now after this, the sterile mycelium, or you can say the sterile hyphae of the mycelium, they start collecting around the sex organs, and they completely enclose the developing asci and ascospores. In this way, by the collection of hyphae of the sterile mycelium, or the sterile hyphae of the mycelium around the asci and ascospores, leads to formation of spherical ball shaped structure known as clistothecium. So it forms a protection around the asci and ascospores. The wall of the mature clistothecium is tough, pseudoparenchymatous, and it is called periderm. In this way, the ascocarp or the clistothecium is developed. Now the asci within the clistothecium remain embedded in the fluid. This fluid is formed by degeneration of inner layer of the periderm at maturity. So when the inner layers of the periderm degenerate, they form a fluid, and these asci, they get embedded in this fluid. Later on, the wall of asci dissolve at maturity, and ascospores float in the fluid within the clistothecium. Then these ascospores are liberated when wall of the Clistothecia break, and when these ascospores fall on the suitable substratum, they germinate to produce a germ tube. So suitable substratum for ascospores is the any vegetable or decaying fruits, because they acquire their food from dead organic matter, and the conditions which are required for the germination of ascospores is the moisture and the favorable temperature. So ascospores will germinate when they get suitable conditions, produce germ tube, and later on form the mycelium. So this is all about the reproduction in case of Penicillium. Thank you.